gosh, it's hard for me to say. I'll tell you the problems I'm working on. One is purely scientific and the other is scientific and romantic. I'll tell you first about the purely scientific. I had a very gifted postdoctoral fellow in my lab, actually two, Eleanor Simpson and Christoph Kellenbach. Christoph is German. We decided we would try to develop animal models of schizophrenia. Now, this is not easy to do because, as you know, schizophrenia has positive, negative, and cognitive symptoms. And the positive symptoms, the hallucinations and delusions, are very difficult to model because how would you know whether the mouse is hallucinating or is delusional? But it struck us that the cognitive symptoms, and even the negative symptoms, are easy to study because a mouse has cognitive capabilities, has a working memory, it uses the frontal lobes. So we decided we would begin by studying the cognitive symptoms of schizophrenia. And we used a genetic approach. There's almost universal agreement that a defect in schizophrenia is increased dopamine in the striatum. This can come from increased release, this can come from increased number of receptors, but a number of different causes. But almost all reliable imaging experiments have shown an increase in dopamine function in the striatum. One genetic cause for this is a particular variant of the D2 receptor, which allows the messenger RNA to persist for longer periods of time. So that leads to an overexpression of the D2 receptor protein. So we modeled this in a mouse. We overexpressed by a slight amount, just as you see clinically, the D2 receptor in the striatum. And my lab had earlier developed techniques for allowing you to express a gene specifically in the striatum, and to have a technique whereby you can shut the gene on and off. So we expressed the gene specifically in the striatum, and we found that you produced a cognitive deficit, very similar to a lesion in the prefrontal cortex, okay, for working memory. And then we turned the gene off, and we found, to our amazement, that the cognitive deficit persisted. So we went back and we found that even when we turned the gene off at birth, the cognitive deficit persisted. What does that mean? It tells you two things. One, it tells you that if you express this gene during development, some other gene gets recruited and compensates, takes over, mediating this deficit. So even when you shut off the D2 receptor, the deficit persists. And we found that overexpression for development leads to an abnormality in the D1 receptor function in the prefrontal cortex. That's number one. Number two, this explains part why D2 receptor blockers don't work for cognitive defects. You give it to somebody who is 15, 18, 20, 25 years old. That's too late. At that time, the striatal D2 receptor is no longer involved. Okay? So this was really very instructive. We then went on to study negative symptoms, and I'll just be very brief. Negative symptoms in people were very interesting characteristic, focusing on motivation as a severe motivational deficit. Motivation has two major components. One is wanting, the other is liking. So a person with schizophrenia, it turns out, likes things as much as you and me. They just don't want to do it. They don't want to spend the energy in doing it. So if you say to them, a schizophrenic person, why don't you come to my house for dinner? They say, oh, it's too much trouble. You live in New York. I don't want to shut myself all the way to New York. I don't want to do it. But if you get them to come to your house for Thanksgiving dinner, they will enjoy the turkey as much as you and me. Okay? And we could show that the mouse is exactly the same. Our mouse, the D2 overexpressor, enjoys the meal or whatever just as much as a wild-type mouse, but it doesn't want to put the effort into it. So now we're collaborating with Ed Smith, and we're trying to do countless studies in people looking at imaging and in mouse models, looking at this particular mouse and other mice in order to have parallel studies in the animal model and in people. So that's one example of the problem I like. The other one is a romantic and scientific problem. This woman who took me to Paris in the first place many years ago told me that Marseille was bored with Marseille was kind of what we see these days. We've been married for 55 years. And she's a major sociologist. She developed the gateway theory of drug abuse. She pointed out that kids don't get involved with cocaine or heroin right away. They begin by experimenting with legally available drugs, like smoking and drinking. These are the gateway drugs. Kids use this first before they go off the hospital. But she could show correlations in a lot of different societies with very well established finding, but she couldn't show causation. So she realized she needed to develop an animal model. She tried to get people to collaborate with her, she couldn't. So I joined forces with her, and we just published a paper in Science Translational Medicine on this. We only explored nicotine so far, we're starting to do alcohol, that if you give nicotine before you give cocaine, the nicotine, the smoking, dramatically enhances effect of cocaine. Dramatically. On the level of behavior, on the level of physiology, on the level of gene expression. If you give cocaine before you give nicotine, it has no effect on nicotine at all. So it's unidirectional. So clearly, a lot of people smoke. Not all of it is cocaine, a very small percent. So she went back and she found the data. Of the people who use cocaine, how many of them are smoking at the time they do it? Practically everyone. And we understand why. You start cocaine without smoking, it doesn't have nearly the effect it has. And if you simultaneously smoke, it really enhances this. So Denise is very pleased. She's always wanted to do biology, and I'm very pleased because we can nice exercise.